Okay, thanks, Fran and Ishi and Felix for the true honour of being the opening speaker for this seminar series. I'm really excited to see what the community is doing. I think it's timely, not just because we're all isolated because of viruses, but as a way of changing how we communicate with each other and thinking about how much jet travel we all need to be doing and how we can still have wonderful interactions with each other using technology as well as face-to-face meetings. And so thanks for taking this initiative. It may have started on a whim, but I'm hoping it takes off and continues. And especially for young people, it's going to be a great way to communicate with each other and talk about ideas and talk about science and be enthusiastic as a community together. So I want to start my talk today by actually doing the acknowledgements, because I tend to rabbit on a little bit, and I want to start with the most important aspect, which is to appreciate the people who've done the work in the lab. Almost all of the experiments I'm going to show you today were done by a single graduate student in the lab, Vicky Stancheva. This is Vicky here. She's, I'm sure, in the audience and would be happy to participate in questions afterwards. Vicky's worked with a number of fantastic collaborators who really helped frame how we think about some of the problems that we're studying, in particular Madan Babu, who's in the Structural Studies Division at the LMB, and his co-workers Shauhan and Balaji have helped us think about intrinsically disordered proteins and domains and how they work and how they might be, as I think Marco and Ishie put it on Twitter, not just linkers, they're the bits that actually do all of the important work. And secondly, we have a fantastic ongoing collaboration with Julia Zanetti and her student, former student Josh Hutchings, who do beautiful electron microscopy and subtomogram averaging, and they've also helped us in thinking about how the coat proteins that we study actually look when they assemble on membranes. So what I want to tell you about today is an unpublished study that is in one form as a preprint on bioRxiv, so you can go and look at the data and some more details about it there. And I'm sort of going to present what I think are the most salient parts and most interesting things that have come out of this study in helping us think about how the COP2 coat assembles vesicles at the endoplasmic reticulum. And so this is the process we study, where the COP2 coat self-assembles on the endoplasmic reticulum, and in doing so, it selects cargo proteins and simultaneously bends the membrane into a spherical transport structure that will then bud off from the ER, travel across the cytosol to the Golgi or to an intermediate compartment, depending on your organism, where this coat must be inherently unstable and fall apart to expose the fusion machinery that will then deliver the membrane and luminal contents to the Golgi upon fusion. And so, as Ishia mentioned, we've known for a very long time what are the components of the COP2 coat. The inner coat is comprised of 3 proteins, a small G protein, SAR1, SEC23, which is the GTPase activating protein for SAR1, and SEC24, which is the cargo adapter that binds to ER export signals and recruits those cargo proteins into the nascent vesicle. And this inner coat is called the inner coat because it also interacts quite intimately with the lipid bilayer in ways that we don't fully understand. It seems to be the very first layer and acts as a bridge between the membrane and the outer coat, which is composed of 2 proteins, SEC13 and SEC31, that have the capacity to self-assemble into a cage-like structure. This is a common feature of other vesicle coat proteins, although the architecture of the SEC13-31 cage is different from other coat scaffolding structures that we know. And so we call this the outer coat because it lies away from the membrane. It doesn't contact the membrane directly, and instead organizes in collaboration with SEC23, SEC24, SAR1 to organize that inner coat layer in some way, collect the cargo-bound complexes, and then ultimately bud off a small spherical vesicle that travels through the cytoplasm. And so there are additional regulatory proteins that also participate in this in very essential and important ways that we still really don't understand. And the confession is we don't understand them because they're really difficult proteins to work with. And so the one that we've studied with a little bit in the lab is SEC16. And in fact, I'm not going to talk too much about that, but these are important regulatory proteins that influence things and make things more complicated. Okay, So this is the cartoon diagram. This is what it actually looks like in a yeast cell. So this is a beautiful electron tomogram that was taken by Alejandro Malera Carrillo, who's a postdoc in my lab, who's worked closely with my most wonderful colleague, Wanda Kukulski. And what Ale did was to hook GFP up to various COP2 coat proteins. And so we know, looking at a fluorescence image, where is a site of vesicle formation, and then we can go in with the electron microscope and pinpoint exactly what are the membrane structures at that site where the COP2 coat is. And so hopefully Ale will get a chance to tell you all about his work in some more detail. I just wanted to show you an example to give you an idea of what the morphology of these things is. Okay, so what I'm showing you here in yellow is the endoplasmic reticulum. We can see these dark ribosomes lined up, making secretory proteins. And we can see a very mature late-stage bud. And this was probably corresponding to something at this stage in the cartoon world. But in the same tomogram slice, if we move away in Z, we can see various other stages of the vesicle budding process. And so this is a relatively early stage bud, or what we imagine is an early stage bud that hasn't yet got the same degree of curvature. And then here is a free vesicle sitting in the cytoplasm. And so we can do segmentation to isolate all of these membranes, and then we can get an idea. This is what the morphology of an ER exit site is. I clicked my mouse too soon. Where we can see all of this membrane remodeling activity going on in a very small patch of real estate on the endoplasmic reticulum. And so the question that I want to talk about today, what Vicky's been studying, is this question at the top here of what governs coat assembly and oligomerization. We know that this outer coat is probably responsible for driving curvature to generate the spherical structures, but we know that it can self-assemble in solution, but we know that it doesn't really do that in a cell. What it does in a cell is self-assemble at sites of ER export, where cargo are concentrated and where the inner coat is assembled. And so we've been really interested in this question of what determines the time and location and organization of the outer coat talking to the inner coat to get this productive vesicle formation event. And so to take a sort of a step back and think about this from a structural perspective, the reason we think that this outer coat actually is the scaffold comes initially from a beautiful single particle cryo-EM structure that was solved by Scott Stagg in 2006, where he took the mammalian Sec1331 outer coat protein and found conditions where they could self-assemble into this spherical cage-like structure. And so the dimensions of these cage-like structures are actually beautifully match the sizes of vesicles that people have seen for decades by electron microscopy. And so the simplest explanation is that Sec1331 can self-assemble at these points of interaction that are called cage vertex interaction sites. And in doing so, it adopts a conformation that can exert force on the underlying lipid bilayer and then bend the membrane and sculpt the vesicle to the dimensions and the geometry that's associated with that cage structure. This all seemed perfectly plausible and even enhanced in probability when beautiful X-ray crystallography from Jonathan Goldberg's lab gave us a really atomic view of what Sec1331 looks like and how it fits beautifully into the cryo-EM density that Scott Stagg saw, where the 1331 has this really beautiful architecture of a tail-to-tail dimer of two Sec13, Sec31 dimers. So this is a dimer of dimers. So the important interactions actually all take place via Sec31. So there's a tail-to-tail dimerization of two Sec31 molecules through these alpha solenoid domains. Then there's a small beta blade that threads through a beta propeller of Sec13, and then a beta propeller structure of its own at either end. So these are the end termini of Sec31 that sit at either end of these long rigid rods. And then when these beta propeller domains come together, or to a point, you get a cage or a scaffold forming. And then the idea is that the intrinsic geometry with which those angles, the angles at which these beta propellers come together, combined with the angles at these sites of interaction, is what determines the size and shape of the underlying vesicle that's formed as that happens. So that was all well and good, but what we wanted to then address is how does that cage assembly event how is that regulated in time and space? And in fact, even more basically, that structural element only comprises about half of the protein. So in this cartoon form, I'm showing you now the beta propeller, which forms those vertex interactions, and this alpha solenoid domain. And you can see that really is only about half the protein. Downstream of those structured domains is a proline-rich region that the field largely ignored for quite a long time. And in part because it's a proline-rich disordered region that makes it difficult to study biochemically and structurally. But we knew that it was important because some fantastic biochemistry and structural biology from Jonathan Goldberg's lab gave us the first clue about what it was doing, where his lab identified what was called the active fragment. So this is a region within this disordered domain that lies across the top of Sec23 SAR1. So this is what I'm showing you here. In pink is Sec23, in green is Sec31, and in beige is SAR1. And this active fragment contains catalytic residues as tryptophan and asparagine that can help drive GTPase stimulation by Sec23 SAR1. And so the idea at the time was that this interface was the site of interaction between the inner coat and the outer coat, and that the engagement of the outer coat would stimulate GTP hydrolysis about tenfold in measurements that had been done by Bruno Antony when he was in Randy Chapman's lab. And that would cause SAR1 to fall off the membrane, and then that would inherently destabilize the rest of the coat, and the rest of the coat could fall off to allow exposure of the fusion machinery. And the importance of that active fragment was hinted at in a functional way by work that Chris Fromme had done in Randy Chapman's lab, again in collaboration with human geneticists who had found a human mutation in this active fragment that changed the interaction and, in fact, the GTP catalysis and the binding interface between Sec31, human Sec31 and Sec23. But it turns out that in yeast, you can delete this entire active fragment and the cells are still perfectly happy. And so that tells us that this active fragment is not the only point of contact or the only assembly interface that mediates the vesicle formation, at least in yeast. But actually then, by this time we actually did this experiment, we weren't too surprised by that, because by then the importance of these triple proline repeats that I'm showing you in the cartoon had been appreciated, again through structural crystallography by Jonathan Goldberg, who found that Sec23 has a gel-solan domain that adopts a fold that looks very much like an SH3 domain, in that it can sandwich triple proline repeats, and that there are triple proline repeats in multiple assembly factors, including Sec31. And the idea then was that you can get by without this active fragment because you also have these sites of interaction of these triple proline repeats with Sec23. And so then we wanted to test the importance of these triple proline repeats, again in yeast. And so Vicky did this in two ways. She made a version of Sec31 that had each of these 7 triple proline repeats that I'm showing here in the cartoon deleted. That was perfectly fine. We worried maybe there are other diprolines with other glycines or alanines that might function instead, so we'll take it a step further and we'll actually make the mutation on Sec23. So we deleted the entire top loop of this gel-solan domain. That's this protein that we call Sec23 delta gel. And when this is the sole copy of Sec23 in cells, they're also actually perfectly fine. And so that tells us that neither the active fragment nor these triple proline repeats are individually essential for inner coat, outer coat interaction for productive vesicle formation and viability in cells. But what we noticed was that if you make these mutations in the context of an N-terminally his-tagged Sec31, now we see lethality. And so let me just take a bit of a detour and explain what we think this means. So in beautiful cryo-electron tomography that Josh Hutchings and Julia Zanetti had done a couple of years ago now, Josh realized that if you use Sec31 that has an N-terminal his tag, so just a 6-hexahistidine tag at the N-terminus, somehow that tag interferes slightly or subtly with this cage formation. So the cage actually can form, and they can see cage vertices in the cryotomograms, but the stability of those interactions seems to be somewhat perturbed. And so they're fewer, or they're less obvious, and so perhaps the interaction interface is just weakened relatively subtly by the presence of that N-terminal histidine. So in yeast, the presence of that N-terminal histidine alone has no phenotypic consequences. It's perfectly fine, and it can assemble onto the coat. The biochemistry is all done and solid. But now in the context of deleting these triple proline repeats, the presence of that N-terminal histidine actually becomes lethal. And so our interpretation of that is that when the cage vertex interaction is perturbed, now the loss of stability that's associated with loss of those triple proline binding interactions becomes untenable. And so we sort of thought to explore that using some biochemical approaches. So what I'm showing you here on the right-hand side is a liposome binding experiment where we take purified COPII proteins, SAR1, Sec23, Sec24, and Sec13, Sec31 in various permutations and combinations of mutations. And we incubate those with small unimillimela vesicles of a defined lipid composition. And then we float those vesicles through a sucrose gradient so that only the bound proteins that are stably associated with the lipid bilayer are recovered at the top of the gradient. And when we do that with the wild type set of proteins, we see really robust recruitment of the entire coat. This is dependent on a non-hydrolyzable nucleotide, so GMP-PNP is required to drive that association. If we have the Sec23 gel solemn domain mutant, self-assembly of the coat is perfectly fine. And it's still fine even if we simultaneously delete the polyprolines in Sec31 and add on that N-terminal his tag. So that tells us that there's the hierarchical binding of the coat one to the other isn't necessarily perturbed by perturbation of these interfaces, even the vertex interactions. And yet, in a cell that is lethal, and in vitro, in a vesicle budding assay from purified proteins, again, but using microsomal membranes from yeast, where now what we're measuring is a release of small, slowly sedimenting vesicles into a vesicle fraction that we can recover and examine by Western blotting for cargo proteins, in this case a protein called ERV46. If you make vesicles, again, using non-hydrolyzable GTP analogues with wild-type protein or with the gel solemn mutant and wild-type Sec31, vesicles form just fine. But when you have this combination of the gel solemn mutant and that destabilized cage vertex, now we no longer see vesicle formation. And so this is really interesting to us because it allows us to separate coat binding. So the liposome flotation tells us just about the hierarchical interactions of the coat. And then something different that has to happen for the coat to actually work to bend a membrane that contains cargo proteins. And so we think that these polyproline sites and the cage vertex interactions then are sort of mutually reinforcing each other and doing work to bend a membrane when membrane bending is a difficult thing for the coat to do. And this is also captured in beautiful EM that Josh and Julia did for us, where they take it's a variant on the liposome binding event, but it uses giant unilamellar vesicles, and instead of looking at binding, it looks at coat tubulation. And so in the wild type condition, Julia showed it beautifully a number of years ago and has fantastic tomographic reconstructions of the coat in these conditions. The coat binds. This is the electron dense coat layer that we can see in these negative stain images, and that the tubes formed from the wild type proteins form these very rigid rod-like structures. And that's because the inner coat layer, as well as the outer coat layer, is very organized. So the inner coat layer forms a helical array that provides structural rigidity and probably contributes to the rigidity and tightness, I guess, of these rods. In contrast, when Josh did the same experiment with Sec23 gel solemn domain mutant and N-terminally his-tagged Sec31, firstly, the number of tubules that were observed was fewer. The coat layer was still observable, which fits with our liposome binding experiments. We're getting assembly of the coat, but the tubules that are formed are not rigid, and we don't see the same degree or the same indication of organization between the inner coats and the outer coats. And so what we think this means is that the hierarchical interaction between the inner coat and the outer coat isn't enough to actually work to bend the membrane and get an organization of the coat that's required for really robust membrane remodeling. And so what we think, this is sort of Vicky's model of how this would work, that Sec1331 has this long intrinsically disordered domain that can come down and sample the surface of the ER membrane where Sec23, Sec24, SAR1 has bound hopefully to cargo molecules and is sitting on the surface of the lipid bilayer of the ER membrane. And it uses multiple sites of interaction, but those are relatively weak and low affinity interactions. But as vertex assembly happens and more of these Sec1331 rods come in, now these relatively low affinity interactions start to self-reinforce each other. And you can bring in more Sec1331, which may bring in more Sec2324, and then do something to help organize that inner coat layer, which is a requirement for actually bending the membrane and getting a productive vesicle formation event. Okay. So I want to sort of then now take a little bit of a detour. Before I finish the rest of the story, I want to take a little bit of a detour and put this into some broader context. And I have to now make a confession that this structure that I showed you earlier, where I pretended that this triple proline repeat was from Sec31, in fact, that triple proline and the crystal structure was generated based on information from a protein, a mammalian protein called Tango1. And so in beautiful work from Vivek Malhotra's lab, Kota Saito identified Tango1 as a pro-collagen ER export receptor and appreciated that a proline-rich domain in Tango1 could interact with Sec23, Sec24. And at the time, Tango1 itself was appreciated to not itself be caught up into these vesicles as they were formed, which is unusual for an ER export receptor. And the model was that Tango1 would bind to 2324 and couple coat assembly to where procollagen was, but then Tango would be displaced by Sec1331, and that would be the mechanism by which Tango could get left behind and procollagen would butt off after this coat had assembled. And so then in the context of these triple proline repeats, okay, just give me a minute. My keynote has quit. Hopefully this isn't fatal. Okay, let's try that again. Nope. Okay. I think it's probably that one slide, which is problematic. And so I'm going to skip that slide and just tell you about it instead. So what that actually, if I'm sharing like this, then you can see it without the playing. So, so when Jonathan Goldberg's lab solved the structure or realized that these triple proline repeats were a site of interaction with Sec23, that then provided this really beautiful elaboration on the initial model where, because it's clear that Tango has multiple polyprolines. And so the idea here is in this cartoon from a review that Josh and Julia published a couple of years ago, where along the length of a Tango1 proline-rich domain, individual polyprolines could interact with different Sec23 molecules, and in doing so would line them up and promote their orientation into this helical array that would then promote the formation of a tube that would be the carrier that could get collagen out of the endoplasmic reticulum. And so I think that this is exciting and has, so what I'm telling you is that what we see in yeast, which obviously doesn't make tubes and doesn't package collagen, beautifully supports this model and tells us that there is more going on than just assembly of the coat, that there is an organizational aspect that has to be fulfilled, even in yeast, where you need an arrangement of the coat that is permissive to bend the membrane and make a vesicle. Okay, so now let's try this and see if now the presentation won't crash. Okay, so about halfway through this project, we were sort of thinking, okay, this active fragment of Sec31 nor the triple proline motifs are individually or in combination actually essential for viability. So there must be another way of interacting between the inner and the outer coats. And so we turned to get some advice from our colleague Madan Babu and his postdoc Xiao Han. who are experts in intrinsically disordered domains. And Xiaohan did this amazing analysis for us where we can plot sequence features along the sequence of Sec1331. And so I'm showing various features here. On the y-axis is the predicted disorder. So above a threshold of 0.5 is predicted to be intrinsically disordered. And you can see, of course, that this disorder matches beautifully with this proline-rich domain, which is a very common feature of intrinsically disordered proteins, that they have a lot of prolines. On top of that, Xiaohan plotted charge effects. And so in grey bars is a total charge for a sliding window of residues. And in red is a net negative charge, and in blue is a cluster of net positive charges. And so what we noticed was that there were clusters of net positive charges all through this proline-rich domain of Sec31, and that these were conserved, that other species had similarly conserved, not positionally, but conserved features of clusters of charged residues, of positively charged residues. So Vicky then went looking on the surface of Sec23 to ask if there was a negatively charged surface that might interact with these positively charged clusters. And lo and behold, so this is a different view. This is a Coulombic depiction of Sec23. So in pink is SAR1. This is the top-down view. So the view that the outer coat would see. In green, again, is the active fragment that lies across the top of Sec23 SAR1. Here's the gel-solin domain that's sticking up towards us, that binding the triple prolines. And here is this great big patch of acidic residues. And so we sort of took the sledgehammer approach and reversed the charge of this region. And so in fact, here now you can see that there's actually a cleft that runs along here. And we just reversed the charges on one side of this cleft, trying not to perturb this interaction with the active fragment. And tested the viability of this charge reversal mutant. So this is a protein we're calling Sec23 delta charge that is completely lethal in cells. The protein is perfectly stable. It binds to Sec24. It binds to SAR1 and the liposome recruitment assay, but it is incapable of binding to Sec31. And so that tells us then that these charge clusters are an important part of the interaction between the inner and the outer surface. And we're now interested in dissecting this a little bit more carefully without this sledgehammer effect of complete charge reversal and probing the precise nature of those charge-based interactions. So I mentioned that these charge clusters were conserved, and so now I'm showing you a comparison of the Cerevisiae Sec31 with human 31A and 31B, where we see these clusters of net positive charges are present in both paralogs. There are different numbers of triple proline repeats, and only Sec31A has catalytic residues that might stimulate GTPase activity. So Madan, who has a strong interest in the evolutionary plasticity of different intrinsically disordered domains, suggested we do the experiment of putting into yeast the human Sec31 disorder domains. I sort of said, no way it's going to work. And that what we should do actually is to put the structural domains interchange, because the structures are actually pretty much identical. All of the cryo-EM and the crystallography has been done on structures that look more or less the same. And so that's a bet. If we'd been betting, I would have lost, because what happened was, if you replace the Cerevisiae intrinsically disordered region with that of human Sec31A, cells are perfectly happy. They're not so happy if you give them Sec31B, and we could talk about why that might be. And they're also not happy, I'm not showing you the data, but they're also not happy if you have the Cerevisiae disordered region with the human structure domain. And so I have lots of theories about that. We haven't explored it more, but I think it's an interesting and exciting thing. So I was very surprised that this unrelated region, so in the last slide, I think I showed the percent similarity. There's 21% similarity with this disordered region in the human Sec31A compared to yeast. We then wondered, how far could we push this? And are there other disordered regions that conserve some of these properties that could completely replace the Sec31 disordered region? And so again, another postdoc in Madan's group, Balaji Santhanam, helped us with this. And so Balaji looked for yeast cytosolic proteins that have multiple polyprolines, so a triple proline repeat and more than 2 or 3 of them. And so this is the sort of the list that he came up with that actually is really interesting, because they fall into what I would call 3 sort of functional categories. There's the COP2 coat, where Sec31 and Sec16 organizational proteins that interact directly with Sec23 via triple proline repeats. there's an actin endocytosis module, and there's an RNA processing module. And so this is just showing the sort of the protein-protein interaction network among these different things. And so we sort of wanted to stay out of the nucleus. And so we thought, let's take some of these actin and endocytosis related proteins, take disorder domains from 4 of these proteins, and we sort of picked them more or less. Well, Xiaohan could probably tell you better than I how we picked them, but he guided us in selecting these. And we chose 4 of them and put just those disordered regions that contain the triple proline repeats into Sec31, so that has the structured domain, and the proline-rich domain has been replaced in this case with disordered regions from any of these 4 proteins. And so, of course, I'm showing you the one that works here, where LAS17 disordered region could completely replace, for full viability, the disordered region of Sec31. And so what's interesting about LAS17 is it has many, many triple proline repeats, and it conserves these charge clusters. It obviously doesn't have an active fragment, so it's not using that mode of interaction, but presumably these other modes of interaction suffice for a robust enough interaction to not only bind, but also remodel the membrane. And so I think that's the key question to us now is, what is the remodeling that's going on when the coat assembles in a productive way versus just assembling in a hierarchical way? And so I sort of want to finish then by saying that what we think the interactions between this disordered region and the inner COP2 coat, it really is, this disordered domain is not just a linker, and it's been overlooked for a number of years, and it's doing the really important parts, and that makes it harder to study, but also makes it really interesting and has some very interesting properties that I think are going to turn out to be important biologically. And so again, really influenced by Madan and how he thinks about disordered proteins and how they work, we think of this now as potentially equivalent to a fuzzy interaction that could be driving vesicle assembly. And so fuzzy interactions use a combination of features to drive biological events. And so they use linear peptide motifs. And so in our case, the active fragment would be considered a linear peptide motif, and for sure these triple proline motifs that have a standard biochemical structured interaction, but relatively low affinity. There are charge effects. And so now, of course, we're interested in the charge mutants that we've made and whether those are a protein-protein interface or more sort of generic or nebulous charge effects like entropic springs or something like that. I didn't tell you about it, but we know that there are length effects. So you have to have a certain length of disordered domain to confer viability. We think multivalency is important because you can knock out one set of interactions as long as you have 2 of the others, then perhaps you're fine as long as you have enough length. So all of these are very common features of fuzzy interactions. And then from the point of the biology of the coat, the importance of these fuzzy interactions is that they can be readily modified by post-translational modifications. And so one of the important features, of course, of all of these coats is they have to bind to the membrane and bend the membrane into a vesicle, but then they have to fall apart. To expose the membranes and to expose the fusion machinery, the coat has to come off. And in fact, we know from the cryo-EM and the cage structures that outer coat scaffold is actually a very rigid and very stable and robust structure. And so I think it's really interesting now to think about how such a robust structure actually is intrinsically unstable and can fall apart very easily. And so what we would like to speculate is that, for instance, in the charge-based interaction, a phosphorylation event by a Golgi resident kinase, of which several have been described, and in fact, beautiful work from Susan Feranovic's lab has defined HRR25 in yeast as modifying all of the components of the COP2 coat, that may then cause charge effects to change and change the affinity of the inner coat for the outer coat and promote the uncoating. And so this is all in the realm of speculation, but I think it highlights some important possibilities about where we might go next. Okay, so with that, this is sort of to depict then what we think is the next stage of what we want to get at is how uncoating happens and whether there are Golgi resident factors that promote uncoating. When does the coat come off? What does that whole event look like? What does the coat look like? What organization actually needs to happen to get a productive vesicle forming when these inner and outer coat interactions are subtly changed? And then how regulatory proteins like Sec16, like Tango1, all of these poorly understood factors that are clearly important to the whole process work. And so with that, I thank you all for coming and listening. You didn't have any choice because I was just talking at you the whole time. This is my first webinar, so it's a different experience, but so I hope you've enjoyed it, and I'm happy to take any questions.